All right, so this is our second lab on cell structures. So I know we're jumping into chapter 4 in our second lab, but that's because chapters 2 and 3 are mostly chemistry, and we don't really do very much chemistry-wise, in labs at least. And so we're going to be moving on to chapter 4. This lab is cell structures, and next lab is going to be mitosis. So we'll have 2 labs for chapter 4. Labs are usually a little bit ahead of lecture, so I guess we are starting out ahead. So first we're going to look a little bit about how cells are studied. Cytology is the study of cells, and we need microscopes in order to study them. This lab we're going to talk mostly about models, but next lab we'll talk a little bit more about microscopes, and you'll have a virtual microscope simulation. And you'll be able to look a little bit more at the structures of mitosis in an onion root tip. So cells are very small. They're measured in micrometers, and we need to use a microscope to view them. And we need to stain them with different staining techniques. And I'll talk a little bit more about staining techniques in our histology lab. So there's a couple different types of microscopes. The most common and the one we'll use is the light microscope. It produces a 2-dimensional images by passing light through a specimen. So this is kind of most common that you've probably used before. Whereas electron microscopes are, a lot more powerful. It beams electrons to illuminate the specimen, and they're also not as commonly used clinically. They have a lot better magnification and resolution, but you'll see them, more in research. There's 2 different types: a transmission electron microscope or a scanning electron microscope. And the scanning electron microscope will give you better 3D images. So this is the traditional light microscope. This is really what we're going to be talking about in this class. And then we have the scanning electron microscope, which looks really cool. You can just see the outside, and this transmission electron microscope where you can kind of see more of the insides. It looks a little bit more like a light microscope, but with better resolution. So our cells vary in shape and size. So an erythrocyte, or a red blood cell, is between 7 to 8 micrometers, whereas a human oocyte, which is the egg, which is the biggest, has a diameter of 120 micrometers. So there's a lot of difference between different types of cells. Most of them are microscopic. Sometimes you can, like the oocyte is just big enough, you could see it with the naked eye. It's the biggest cell, but most of the rest of them are going to be microscopic. But the shapes of our cells are vary. Some of them are spheres, some of them look like cubes, some of them look like columns. We'll talk about these more in our histology lab in a few weeks. Some of them are disc shaped, some of them are irregular and look like spiders. Those are going to be our nerve cells. In this class, in this section, we're just going to kind of talk about a general cell, and we'll imagine that it looks round, but know that they can have lots of other different shapes. And so here is going to be most of our cells that we're looking at. This is the mitochondria, which is an organelle within the cell. Bacteria is pretty small, and then the human oocyte is the biggest. And so we can see, like the unaided eye, we can just see the oocyte. And then under here, we have all the smaller things like viruses and ribosomes and atoms and molecules that we've been talking about so far. Large macromolecules and proteins are going to be over here. So obviously cells are bigger than proteins and bigger than atoms, but smaller than tissues. If we go back to our organization of life. So as I said before, there are lots of different types of cell shapes. So the nerve cell looks kind of spider-shaped. Red blood cells are these biconcave discs. When we look at epithelial tissues, a lot of them are either cube-shaped or cuboidal-shaped or columnar-shaped. Our cartilage cells can look kind of spherical, and these skeletal muscle cells are long cylinders. So we'll talk all about these different cells when we start talking about histology. But for this class, at least for this lab, we'll just kind of think about a generalized cell, and we'll imagine that it's circular. But just know that it can be all these other different shapes. Now, the 3 main features of the cell, 3, that we can see are the plasma membrane, the nucleus, and the cytoplasm. And so generally, if you look at a cell under, fairly low magnification, you're going to see the outline, which is the plasma membrane, and then you're going to see the nucleus in the middle, which stains dark. And everything in between the plasma membrane and the nucleus is going to be the cytoplasm. So the plasma membrane is made of phospholipids. Remember, we've talked about phospholipids already. They are those amphipathic molecules with polar heads and nonpolar tails. This is going to be really important because the plasma membrane is going to make a barrier that separates the inside of the cell from the outside. Our plasma membrane can have some special extensions called cilia, flagella, and microvilli, which we'll talk about at the end of this presentation. The nucleus is going to be the largest structure in the cell, and it's going to be in the middle, and it stains pretty dark. It's enclosed by a nuclear envelope, and the nuclear envelope is just a double layer of this plasma membrane stuff. So it's also made of a phospholipid bilayer. It contains the genetic material, or DNA, and then all of the fluid inside the nucleus is called the nucleoplasm. And then everything in between the plasma membrane and the nucleus is called the cytoplasm. And so at low powers with just a light microscope, a lot of times you cannot make out very many features of the different organelles. It just kind of looks like a blur. And so we call all of that the cytoplasm, and it includes the cytosol, which is the fluid inside the cell, the organelles, and any inclusions, which are like temporary storage things. So what exactly is the cytoplasm again, right? We said the cytosol is the intracellular fluid, and this is the viscous stuff in the cytoplasm. It has a really high water content, but it also has dissolved things like glucose and ions, and we'll talk a lot about that as we go through this course. Organelles means little organs. So they're the little tiny things inside the cell that kind of work like organs to help the cell, do things like maintain its energy, or obtain nutrients, or get rid of wastes. They are complex and organized structures. They each have unique shapes and functions, and we're learning all of them today. So there are two categories. The membrane-bound organelles, these are surrounded by a plasma membrane of a phospholipid bilayer. So it's just the same type of bilayer that surrounds the nucleus and surrounds the outside of the cell. And the nice thing about these membrane-bound organelles is that this membrane is going to let them have a different internal contents than the external contents. So they can have, like the lysosomes can have digestive enzymes that are inside them, and then they won't be able to escape and harm the rest of the cell. And then finally, we have non-membrane bound organelles. These organelles are generally made of protein, and they are not surrounded by a membrane. So membrane-bound organelles are going to include the endoplasmic reticulum, both rough and smooth, the Golgi apparatus, the lysosomes, the peroxisomes, and the mitochondria. And then non-membrane-bound organelles are going to include ribosomes, the cytoskeleton, the centrosome, and proteasomes. Inclusions, as I said, are temporary storage materials. They generally are going to be things like pigments, so like melanin, glycogen, or triglycerides. So both of these are energy storage. So glycogen, remember, is made up of glucose, and triglycerides are the storage form of fats. So here's our generalized structure of the cell. Make sure you can label this and you know what all of these things are. APR is also your friend. Make sure you look through APR. And if you have done the introductory, like last week's assignment, for last week's lab assignment, talked you through how to use APR. So just go to the structures of the cell and look at the different parts of the cell and the plasma membrane on APR. Your book also has a lot of good pictures like this. So the general functions of our cells that we need all of these organelles to help perform are: the cell needs to maintain integrity and shape. This is dependent on the plasma membrane, especially, so we'll talk about that and what it looks like. The cell also needs to obtain nutrients and form chemical building blocks. And it needs the mitochondria to harvest energy for survival, so the mitochondria is really important in getting energy. It needs to dispose of wastes, and so the lysosomes are going to be important, as well as the peroxisomes for disposing of wastes. And then some are capable of cell division, and that's going to make more cells of the same type, and that helps maintain the tissue by providing cells for new growth and replacing dead cells. We're going to talk about cell division in the next lab. So we're going to start with the plasma membrane and kind of work our way down and work our way into the cell. So the plasma membrane is the outside of the cell, right? And it's made up of about equal parts lipid and protein by weight. So the lipid portion, as we said, are those phospholipids, that phospholipid bilayer. And then we have several different types of proteins in there too. The plasma membrane is really important because it regulates the movement of substances into and out of the cell. And we're going to have an entire very long lecture discussion about how it does this. So a couple of different types of lipids that are included in the plasma membrane are phospholipids, which we've talked about a few times now, cholesterol, as well as glycolipids. And so phospholipids, remember, are amphipathic molecules. They have a polar head and two non-polar and hydrophobic tails. And so they're going to form two parallel sheets of molecules that lie tail to tail, right? So remember, the tails are going to hide on the inside, and the hydrophobic heads are going to be on the outside, directed outward towards the water. The hydrophilic polar heads directed outward towards the water. And the phospholipid bilayer is kind of the thing that makes up the backbone or the framework of the plasma membrane. And it makes sure that the cytosol, which is the intracellular fluid, remains inside the cell, and that the interstitial fluid, or the extracellular fluid, remains on the outside. Cholesterol is going to be scattered within the phospholipid bilayer. Cholesterol, remember, is a type of steroid, which is a type of lipid. It's got the ring structure, the classic ring structure. It helps strengthen the membrane and stabilize it against temperature extremes. And we can also use cholesterol to form like steroid hormones and things. And then finally, glycolipids are lipids with attached carbohydrates groups, right? So glyco, like think glycogen, that's going to mean carbohydrate, and then lipid is going to be a lipid that has a carbohydrate attached. They're located on the outside of the phospholipid region only, and they help form what's called the glycocalyx, or the sugar coating of the cell. And so we're also going to have some glycoproteins that help form that. So the other thing that makes up our plasma membrane are going to be proteins. And proteins are about half of the plasma membrane by weight. They actually float and move throughout the fluid bilayer. And they perform most of the functions of the membrane. We said they are going to be important for, they can be enzymes, they can be receptors, they can be channels. So there's a lot of different things they can do. There are two different structural types called integral proteins and peripheral proteins. So integral proteins are going to go all the way through, and they go from one side of the plasma membrane to the other, whereas peripheral proteins either float on the inside or the outside of the plasma membrane. And so we can see in this picture here, make sure you can identify all the stuff in this picture. So the phospholipid heads right here in purple, and then the little tails are yellow, so those are the phospholipids. These little yellow guys here with this ring structure are the cholesterols. The blue ones are the proteins. The ones that go all the way through are integral proteins. The ones that sit on the outside or the inside are the peripheral proteins. And then if anything has a carbohydrate attached, that's going to be a glyco something, right? So the carbohydrate attached to the lipid is a glycolipid, and the carbohydrate attached to the protein is the glycoprotein. And so, as we said, the integral proteins are going to extend across the bilayer. They have hydrophobic regions on the inside that interact with the hydrophobic tails, and the hydrophilic regions are on the outside, and they're going to go on either side of the membrane. And a lot of them can be glycoproteins. The peripheral proteins are not embedded in the lipid bilayer, but as I said before, they kind of float on the surface, on the outside or in the inside of the cell. Now, I'm going to talk about some categories of proteins, and there's 6 different categories of proteins. And so the first one is going to be these transport proteins here. These are going to be really important, and we're going to talk about these in class. And transport proteins are going to regulate the movement of substances across the membrane. So channels, carriers, pumps, symporters, and antiporters. So we'll have a long lecture about all of that. Cell surface receptors are molecules that can bind things that are called ligands. And a ligand is just a chemical messenger that's going to bind to those proteins. So examples of ligands would be neurotransmitters from the nervous system or hormones from the endocrine system. And so in this picture here, we can see a cell surface receptor is going to be a protein that binds to a specific ligand, and it's usually going to cause some sort of change inside the cell. We can also have identity markers, and these identity markers are usually going to be those glycoproteins, right? And they make up what we call that glycocalyx, or the sugar coating of the cell. And so these are used to distinguish healthy cells from unwanted cells or cells to be destroyed. They communicate to other cells that they belong to the body. And we'll talk a little bit more about that in the immune system in 169. The next structure is an enzyme, and enzymes are going to be covered extensively in chapter 3. So these enzymes are going to catalyze chemical reactions and make them go faster. And so they can be attached either to the outside or the inside of, or they could be integral proteins or peripheral proteins on the outside or the inside of the cell. But they're just going to make these reactions happen faster. We can also have an anchoring protein. So anchoring proteins are going to anchor the cytoskeleton, which is one of the organelles inside the cell. It's kind of like the bones of the cell. It's going to anchor it to the plasma membrane so that the cell can keep its shape. And then finally, we have these cell adhesion proteins. And these proteins are going to join two neighboring cells together. And we'll talk about some different types of these proteins in the tissues lab. All right, so that was the plasma membrane. And next we're going to talk about the nucleus. So the nucleus is going to be very complicated, and the outside of the nucleus is going to be surrounded by the nuclear envelope. The nuclear envelope is a double layer of phospholipid. So it's like a double phospholipid bilayer. So it's like 4 layers of phospholipids that encloses the nucleus. It separates the cytoplasm from the fluid within the nucleus, which we said was called the nucleoplasm. And on the outside, sort of the outer layer of that is continuous with the rough endoplasmic reticulum. So it kind of turns into the rough endoplasmic reticulum. There are openings in the nuclear envelope called nuclear pores. And these open passageways are formed by some proteins, and they let bigger things into and out of the nucleus. So it's like messenger RNA, as well as ions and water-soluble molecules. They can actually just pass through the nucleus, but pass through the nuclear pores. Within the nucleus, we have something called a nucleolus. And a nucleolus is a dark, small, dark staining thing in the middle of the nucleus. It is not membrane bound, and so it doesn't have a membrane around it. It's more made of protein as well as RNA. And it makes ribosomes. It produces the small and the large subunit of ribosomes. Ribosomes are going to be important in making proteins. These nucleoli are not present in all cells, but if the cells need to make proteins, you'll have more of them. So you might have more than one in nerve cells because it needs to make a lot of proteins, but we do not have any nucleoli in sperm cells because they don't need proteins. So the number of nucleoli are going to differ depending on how many proteins that cell needs to make. The more proteins it needs to make, the more nucleoli it might have. So this picture shows us, this is that double phospholipid bilayer, that's the nuclear membrane. These holes are the nuclear pores. And here it would continue out and be continuous with the rough endoplasmic reticulum. On the inside we have the nucleoplasm, and all of this stuff in here is going to be filled with chromatin, which is this uncoiled form of DNA. And then this dark staining body in the middle is the nucleolus, right, which is going to be making those ribosome subunits. So now we're going to move on to the membrane-bound organelles. And our membrane-bound organelles, as we said, one of them is the rough endoplasmic reticulum that's literally connected to the nuclear membrane. And all of our membrane-bound organelles are surrounded by a membrane, and these membranes are going to be made of phospholipid bilayers. So phospholipids are really important. It allows an isolated environment inside those organelles in order to perform different chemical reactions. The first one we'll talk about is the endoplasmic reticulum. And this is going to be, we have 2 different flavors of it. A rough ER has ribosomes attached to the outside, and the smooth ER does not have ribosomes. This compounds about half of the membrane within the cells, and they both have different jobs. So here is the rough ER. We can see it's continuous with the nuclear envelope, and it's covered in ribosomes. And then the smooth ER over here looks similar, but does not have ribosomes. As you can see, it's kind of attached over here to the rough ER. It does not have ribosomes. So the rough ER is very important in producing proteins, right? Because the ribosomes are embedded in it, and ribosomes make proteins. And so the ribosomes make the proteins, and then the proteins actually go into the rough ER, and the rough ER is going to modify that protein. So it's going to modify it and get it ready for shipping. And then it's going to transport it out of the rough ER. And the transport vesicles, which are these little areas, these little tiny baskets of, they're empty, they're surrounded by a phospholipid bilayer, and they're carrying those proteins. they transport vesicles shuttle the proteins from the rough ER to the Golgi apparatus. So they're going to transport out to the Golgi apparatus. So the rough ER is going to take the proteins that are made by the ribosomes, change them and get them ready, and then it's going to ship them out to the Golgi. The smooth ER does a little bit different. It has a lot of metabolic functions, and it might be different cell by cell, but some of the functions are to synthesize, transport, and store lipids. So this is a good one to know. The rough ER does proteins, the smooth ER does lipids. It can help with carbohydrate metabolism, and it also is important in detoxification of drugs and poisons, right? So our liver cells, because the liver does a lot of detoxification of our blood, is going to have a more extensive smooth ER. So we said that the rough ER transports things to the Golgi apparatus. The Golgi apparatus is another membrane-bound organelle, and it has these sort of sac-like membranous structures. It exhibits what we call polarity, which means it has two different sides. It has what we call a cis face, which is close to the rough ER, and a trans face, which is far from the rough ER. And so this is going to receive the proteins from the rough ER, and then it's going to modify the proteins, and then it's going to ship them out from the trans face. So it modifies, packages, and sorts the proteins and gets them ready for export. The Golgi apparatus then, instead of making a transport vesicle that just transports it within the cell, it makes a secretory vesicle. And these vesicles are going to generally become part of the plasma membrane or release the contents outside of the cell. So they're going to go merge with the plasma membrane. So here we've got the rough ER. This is the cis face of the Golgi apparatus. It receives the packages from the rough ER. It modifies those proteins, and then it's going to sort them, and the trans face is going to ship them out. And these secretory vesicles are going to go ahead and merge with the plasma membrane to release those proteins outside. This is called vesicular transport, and we're going to talk more about this in lecture when we talk about plasma membrane regulation of materials moving into and out of the cell. Some of these, instead of shipping them out, some of these can actually become lysosomes. So we'll talk about lysosomes in just a second, right now. Lysosomes are small membranous sacs that contain hydrolytic digestive enzymes. And so these are going to destroy like unneeded parts of the cell. And they can also, if we have like the cell is a phagocytic cell and it eats up some sort of bacteria, then it's going to bind that bacteria with the lysosome, and the lysosome can go munch up that bacteria and digest it. So this is digest the contents of those endocytosed vesicles. So the lysosome is kind of like the stomach of the cell. It digests things. Peroxisomes are similar to lysosomes. They're a little bit smaller. These, instead of coming from the Golgi apparatus, they come from the rough ER. So these peroxisomes come from the rough ER, and they are going to have a couple of different functions. So they break down molecules like fatty acids, amino acids, and they make hydrogen peroxide, and they use hydrogen peroxide to do this instead of hydrolytic enzymes. But they also form specific type of lipids like bile salts. All of these membrane organelles are going to participate in what's called the endomembrane system. So this is like the membrane system within the cell. This includes the smooth and rough ER, the Golgi apparatus, the vesicles, the lysosomes, and the peroxisomes, as well as the plasma membrane and the nuclear envelope. It does not include the mitochondria. The mitochondria is the only one that doesn't participate. It provides a means of transporting substances within the cells. So all of these are covered by these phospholipid bilayers. And so we can have these little transport vesicles that bud off of one organelle and then go into another organelle. And so they can all sort of share their phospholipids and transport things from one organelle to the next. But the mitochondria does not participate in this. The mitochondria is kind of its own thing. It has its own different, it has an outer mitochondrial membrane and an inner mitochondrial membrane, so it looks a little bit different than most of the other organelles. And it's really important because it's the powerhouse of the cell. It is going to produce ATP that's needed for the cell. And if we look here, I do want you guys to know a little bit about the structural organization of the mitochondria. So the outer mitochondrial membrane is this outer layer over here. The inner mitochondrial membrane is going to be this guy here that has these little folds in it. The folds are called the cristae, and then we have the matrix on the inside of the mitochondria. And we'll talk more about this when we get to the mitochondria in chapter 3. So as we can see, the matrix is right here, kind of on the inside. So that was all of our membrane-bound organelles. And so all of our membrane-bound organelles except for the mitochondria participate in that endomembrane system. But now we're going to talk a little bit about the non-membrane-bound organelles. So the first one is a ribosome. And we already said that the ribosomes are going to make proteins, but they're actually made out of proteins also. So they contain proteins and a ribonucleic acid called an rRNA that make up these ribosomes. There's a large subunit and a small subunit that kind of sit together. And they're made within the nucleolus, but they're assembled in the cytoplasm. And there are two different types of ribosomes. One of them are bound to the outside of the rough ER. These are going to synthesize proteins that are going to be exported out of the cell, or they're going to become part of the plasma membrane, or maybe they'll serve as enzymes in lysosomes. Whereas our free ribosomes are suspended within the cytosol, and they make proteins that the cell is going to use. So like used inside the cell. And our ribosomes can actually like start in the cytosol and bind to the rough ER and then unbind too. So they can kind of move around. So here's our ribosome. We have the large subunit and the small subunit. They form this big ribosome together, the functional ribosome, and then this is going to be really important in translation of protein, so of messenger RNA into protein. And we can see on here we have some ribosomes bound to the rough ER and some that are free in the cytoplasm. Next we have the centrosome. Centrosome is a pair of centrioles. So we have a pair of organelles called centrioles. They are going to basically organize the microtubules within the cytoskeleton. And I know we haven't talked about the cytoskeleton yet, but we'll get there. They're also really important in cell division, which we'll talk about next week. So the centrosome is made of two centrioles, and each centriole is made up of triplets of microtubules that are kind of arranged in this little thing that looks kind of like a Twizzler. And so they're going to kind of oversee all the microtubules in the cell, as well as form the mitotic spindle during mitosis. Proteasomes, we can't see the proteasomes on the models that we have in lab, but they are protein digesting organelles. And they're located in the cytosol and the nucleus. They're going to degrade cell proteins. They do need ATP to do this. And they only are going to break down proteins that are marked with a specific tag, the ubiquitin tag. And that way, they're going to kind of recycle the amino acids so we can use them for something else. As they age, they might start not working as well, so they may not be able to get rid of those proteins as well. So this is what it looks like. This protein has to be tagged with this ubiquitin molecule, and then the protein is going to kind of go through this little guy. I imagine it sort of chopping up the amino acids, and then these amino acids can be recycled by the cell. And it's also going to make sure that the cell is only going to be making good protein. So any like misfolded or faulty protein is going to get broken down. Now we have the cytoskeleton. So the cytoskeleton is going to play a role. It's like the bones of the cell. It makes sure that the cell has support, but it also helps move the organelles around. They're important in cell division and moving other materials around inside the cell. It's going to be found all in the middle and the inside of the cell. It's going to anchor to those anchor proteins in the cell membrane that we talked about. There are three main types to know. The smallest are the microfilaments. The middle are the intermediate filaments, right? That looks like middle. And then the biggest are the microtubules. So microfilaments here are the smallest, and they are actin protein monomers in two twisted filaments. And so these are going to be important for maintaining cell shape, internal support, and cell division. And remember, actin is going to be a type of protein that we also see in muscles that help the muscles contract. Intermediate sized filaments are a little bit bigger and more rigid than microfilaments. They help with cell-to-cell junctions and cell support, and different types of cells are going to have sort of intermediate filaments that are slightly different, made of different things. Microtubules are the largest, and they're composed of something called tubulin. And so it kind of looks a little bit like these long straws, and they can be made longer or shorter as needed. This is important for maintaining shape, transporting things within the cell, as well as cell division. So this picture shows this little actin microfilament here. It's the smallest. The intermediate filament is in the middle, and the microtubule is the biggest. And I think it looks a little bit like a tube. It looks like it has a hollow tube in the middle. Now finally, I said that the outside of this plasma membrane can have some specialized structures. And I guess maybe I should have talked about these then, but here we are. So the outside can have 3 different types of specialized structures: cilia, flagella, or microvilli. So I'm going to start with the smallest and work my way up. The microvilli are the smallest. These are tiny. They're extensions of the plasma membrane that increase the surface area of the cell, but they do not move on their own. The next middle one, this is the intermediate sized one, is the cilia. Cilia are hair-like projections. They're bigger than microvilli, but they kind of look the same. They're just longer. But they actually can move under their own power, and so they're going to move things along the cell surface. So they don't move the cell themselves. The cell stays together, but they beat and move things along the cell surface. Then finally, the largest is the flagella. This is longer and wider than the cilia, and it actually propels the entire cell, and the only human cell to have one is the sperm cell. So this is a picture of the microvilli. This is the smallest one. The cilia would be a lot bigger. And on APR, you can see they have some cilia and microvilli that you guys can compare one to the other. All right, so don't forget to look at APR and watch the videos for this lab, and let me know if you guys have any questions.